Every year, Philippine temperatures continue to rise and typhoons intensify with global warming. In 2024, the country will have reached dangerous heat indexes, and an average of 20 typhoons enter the Philippine area of responsibility per year. These leave a lasting impact. In the past decade, our agricultural sector has procured about 290 billion pesos worth of damages. As such, we need more innovations and strategies to support millions of Filipinos by equipping ourselves with the means to combat climate change. Now, what if I told you that we could use DNA, the blueprint of life that codes for the rules of what makes up humans and other species, by editing it so that we can give plant DNA the ability to resist floods, high temperatures, and droughts? Well, we can do that now with CRISPR-Cas9. To understand how groundbreaking it is, we would have to know that altering genetic material is challenging since DNA is highly sensitive. A single change in its code may or may not alter the meaning of the entire sequence. Back then, we used DNA pellets, which could only be inserted manually into the DNA of a cell. It is a trial and error phase. Otherwise, you need to exhaust plenty of resources. With CRISPR-Cas9, you can target a specific region of DNA that you want to change, which is way more efficient than hoping for bullets to hit the correct sequences. But how exactly does that work? It all started with the discovery of CRISPR. Scientists have found that a specific DNA sequence in bacteria can function as an immune response against bacteriophages. These viruses insert their DNA into the bacterium and steal their cellular machinery. This allows their DNA to replicate and thrive in the cell. Now, when the bacterium survives an invasion of this virus, they have a protein complex that can gather data of the viral invader's DNA. The complex can do this by cutting and encoding them into the CRISPR sequence for storage. This is important because if the bacteria is attacked by the same type of bacteriophage in the future, it can get the viral information stored in the CRISPR sequence by decoding it into a dual RNA system. This RNA can then be used by an enzyme, Cas9, as a copy of the viral DNA sequence. With this, Cas9 can now correctly identify the bacteriophage and slice them off cleanly with precision during the next invasion. Scientists Jennifer Duwadna and Emmanuel Charpentier were able to make Cas9 target other DNA sequences, like those in animals, plants, and even our own human DNA. They did it by altering the single guide RNA to match a DNA sequence you want to edit, so that the Cas9 protein can use it as a reference to target your desired sequence. With this, you can do plenty of things with CRISPR. You can delete regions of DNA you don't want by targeting the ends of the undesired strands. You can also add enzymes to this complex, which can alter the nucleotide basis of DNA. You can even enhance genes by incorporating transcription factors that can enhance certain traits. Because of CRISPR's powerful targeting mechanism, it holds almost limitless potential in genetic engineering.
Now that we understand how CRISPR works, let's see how we can use them to address agricultural challenges. Plants have specific genes involved in stress response that helps them become more resilient against environmental stress. Researchers target these genes using CRISPR, and they can edit them by altering sgRNA that the Cas9 uses as a reference for detecting which sequences to modify. Afterwards, this system is cloned and applied to the plant through transformation. Once the plant has been regenerated, the edits are confirmed, and these genetically modified plants undergo phenotyping and field trials to test their application. Now that we know the plant editing protocol, how do we exactly apply the knowledge in an agricultural context? Starting with drought, a natural enemy of food crops. One study found that when a specific gene called Arab1 is more active, plants become more drought tolerant. But if this gene is reduced, the plants become more sensitive and struggle without water. Another study looked at plants' many built-in systems to handle stress. One important gene is SIMAPK3 helps protect plant cells from damage when water is scarce. Without it, plants show signs of stress like wilting, more internal damage, and fewer protective antioxidants. It doesn't stop there. Ultimately, researchers have found that removing a gene linked to the seed growth of a tomato plant called SILBD40 actually makes the plant stronger in dry conditions. Even more good news for tomatoes, another gene CRISPR normally helps plants respond to a hormone called auxin. But when the scientists turned off this gene, the tomatoes handled drought much more better. And so, from boosting helpful genes to turning off the ones that makes plants weaker, researchers are finding amazing ways to help crops grow even when the rain doesn't fall. Salt in soil is a growing concern for farmers worldwide. It can prevent plants from absorbing water, disrupt their growth, and even hinder seed germination. But thanks to CRISPR, scientists are finding ways to help crops survive in salty conditions. In recent years, researchers have used CRISPR to edit the genes of crops like rice and soybeans, making them more tolerant to salt. These changes allow plants to better manage the salt level inside their cells. For example, certain edited genes like OSRR2, OSDST, and many more help the plants move salt around more effectively, boost stress signaling, and protect cell functions. Another study from 2023 showed how these gene changes help crops stay healthy even in salty soil. The plants grew better, sprouted more easily, and produced well without losing the traits farmers depend on. This technology could make a difference in areas where poor irrigation or climate change has left soils too salty to farm. As temperatures rise due to climate change, heat stress has become a major threat to crops. Extreme heat can damage the parts of a plant that produce energy, especially in important food crops like rice and wheat. When it gets too hot, the plant's ability to turn sunlight into energy called photosynthesis slows down or even stops. But again, CRISPR is there to help. Scientists are now using this gene editing tool to make crops more resistant to high temperatures. By changing certain genes in rice, they found ways to protect the parts of the plants responsible for their energy production. This keeps the plants working properly even during heat waves. These CRISPR changes don't just protect plants from heat damage, they also help crops keep growing and producing food when the weather is extreme. It's an exciting step towards making our food systems more stable in a warming world.
Despite all of the progress that has been made, CRISPR still faces some challenges in its delivery and efficiency and potential off-target effects. Let's dive into how these affect CRISPR research. First, delivery limitations. In order to produce the desired results, CRISPR-Cas9 has to first be successfully delivered to the cells. However, this is difficult to achieve because it requires all of the components to be delivered into the cells at the right concentrations and at the correct point in a cell's cycle. Another limitation to consider is that even if the cell takes up the CRISPR-Cas9 complex, the gene editing activity may not even occur. This is more apparent when trying to knock in or insert material into a gene. Lastly, off-target effects. This is when gene editing occurs in an untargeted area of the genome, leading to unwanted results. This is a major concern in experiments using CRISPR-Cas9 due to its potentially hazardous effects. To combat this, researchers have made efforts to improve in silico off-target prediction tools and reduce off-target effects by improving the Cas9 nucleases. In the matter of genetic innovation, the research on hybrid rice conducted by the International Rice Research Institute holds transformative potential for medicine and various future applications by using advanced breeding techniques that accelerate the development of hybrid rice varieties. Achieved using a variety of genomic modifications, the methods have improved crops that are designated to withstand challenges such as drought, flooding, salinity, and to emit lower levels of methane. Since the strategic management of our lands and crops is crucial in this climate crisis, the high-yield hybrid varieties are significantly advantageous due to their shorter growth durations, which could benefit the rice paddy's sustainability. They are paired with strategic innovations such as optimized fertilizer use, controlled irrigation, and effective organic matter management can minimize climate change, allowing a dual approach for food production and global warming.